So that was a great talk. Raj hasn't slept. So that was amazing that he was able to keep that up. Okay, let's move on to another great speaker. Dr. John Lee is an assistant professor of medicine at the Brigham and Harvard Medical School. He's a translational HIV researcher who focuses on HIV persistence, reservoirs, and post-treatment control. He's also the associate director of the clinical core for the Harvard CFAR and the director of the Harvard Virology Specialty Lab for the ACTG. Welcome, John.
All right, thank you, Pasola, and it's really heartening to see so many people in the community be engaged in this process and be interested in learning about cure research, because without all of you guys, none of this work could be possible, and just really happy to see it, and I'm glad to be here today. My charge today was to talk about HIV treatment interruption. It's a topic of increasing interest and importance as more clinical trials and start using this, and especially since we have spent 2 decades telling our patients not to ever stop treatment. So the question that we get is, why now, and how will this help HIV cure research, and how can we do it safely, especially since up to now most trials don't use treatment interruption, but I would say more and more trials do now. Before we knew whether or not Raj could actually make it from his flight over here today, I had put in some slides on background on HIV cure, which I think we can move through relatively quickly since he's given such a clear description of the field. And the overview of the talk is that we'll start off by talking a little about an overview of the search for a cure. I'll just very briefly go through the Berlin patient and the importance of treatment interruption trials. And then talk a little about the risk of treatment interruption studies, as well as the ethical considerations. And then I just want to give a plug on my own study, which is an AIDS Clinical Trials Group study 5345. So again, a summary of the kind of the global HIV epidemic. There are now, and this is actually data from a couple years back from the WHO, that there were at that time 37 million people infected with HIV, and in 2016, there were 1.8 million new infections and 1 million HIV-related deaths. So we know that antiretroviral therapy has been an incredible success story, but there is so much work left to be done. And I think most of us believe that in order that an HIV cure is really needed in order to bend the curve of the epidemic and to really bring the epidemic hopefully to a close. And as Raj alluded to, our patients can be on treatment for months, years, or even decades. But as soon as they stop therapy, HIV almost always will rebound, and actually rebound relatively quickly. And the reason this happens, again, is because HIV persists within the cells. And this is, I'm showing you a graph in a cohort of individuals who were treated for over 10 years. And the lines show the levels of HIV DNA. And by measuring HIV DNA, it's a reflection of the size of the reservoir, okay, level HIV DNA. And what you can see here is that in the HIV DNA, or the reservoir, decreases most dramatically in the first year after antiretroviral therapy is started. That between years 1 and 4, the reservoir decrease kind of starts leveling off, and then really the size of the HIV reservoir doesn't really decrease too much after about 4 years or so, showing that HIV continues to persist within our patients' bodies despite long-term antiretroviral therapy, and that we cannot fully eradicate the virus. Now, is an HIV cure possible? As Raj has already mentioned, there has been at least one case that I think we can fairly confidently say that a cure has been achieved, and this is Timothy Ray Brown, or the Berlin patient. And in order to understand how he was able to be cured, it's important to, again, show that HIV, which is seen by this kind of cartoon here, binds to 2 different co-receptors, a CD4 receptor as well as to the CCR5 co-receptor, and that there are individuals who have this deletion in their CCR5 receptor. There's something we call CCR5 delta 32 because they have a 32 base pair deletion in the CCR5 receptor that makes it non-functional. And because of that, these individuals, when they have a deletion in both copies of their CCR5, are actually functionally resistant to HIV infection. And for Timothy Rebound, he unfortunately had to go through a diagnosis of acute myeloid leukemia. Right? So he actually had AML diagnosis, and this graph shows his viral load over time. He had an interruption while he was undergoing chemotherapy, and you can see his viral load rebounded here to relatively high levels. And then it came down after he restarted antiretroviral therapy. And then he underwent what's called a stem cell transplant, right? So during a stem cell transplant, what happens is that the cells from the patient or the recipient, first of all, are killed during conditioning chemotherapy as well as irradiation. And then these patients then receive donor stem cells from someone who is healthy, and which then over time will replace the cells from the patient or the recipient, such that their cells are now completely replaced by the donor cells. And in this case, as Raj mentioned, the donor cells were missing the CCR5 co-receptor, such that these donor cells were actually inherently resistant to HIV infection. And after he received this stem cell transplant, he stopped antiretroviral therapy, and the virus remained suppressed. And he ended up having a relapse of his cancer, ended up needing a second stem cell transplant. And it's been now more than 10 years, actually now more than 11 years, and he has remained HIV-free without antiretroviral therapy. Now, as we've heard, stem cell transplants or bone marrow transplants have a really high, 10 to 25% upfront death rate. So this is something that you would only receive if you had, life-threatening cancer, and not really something that we can use for all of our patients on a routine basis. And so, of course, what I'm trying to say is don't stop taking your medicines at home, and that this is a very special circumstance. But I do want to mention that without treatment interruption and trying Timothy Rebound off treatment, we would never have known that he was cured, right? That while there are a lot of clinical trials right now that don't use treatment interruption. They look at things like, can it affect the size of the reservoir, or can it boost a certain immune response that they're looking for? If you really want to evaluate whether an intervention can lead to a cure or HIV remission, in the end, you can only judge the ability of these interventions by doing a treatment interruption trial, either by showing that such an intervention can delay viral rebound or can significantly reduce the viral load set point. And I wanted to give you an example so that we're all on the same page of what a treatment interruption trial looks like. So this is a paper that was just published last year by a group led by Tony Fauci. You might have heard of him. He is the head of the NIAID, or the National Institutes of Allergies and Infectious Disease. And he did a trial looking at a particular therapeutic vaccine. This vaccine had been shown to be immunogenic, which means that it in prior studies has been shown to boost the immune response. And now what they're trying to see is, can it help delay when the virus came back after treatment interruption? And so at the beginning, participants were split into two groups: participants who received the vaccine or those who received placebo. And that happened in the first part, and the arrows show when they received the vaccine or the placebo. And then the kind of the orange area here is the time frame where these individuals underwent a treatment interruption when their therapies were stopped. And what they looked at is, when did the virus come back? And did the vaccine help delay the virus rebound time? And and here is a graph showing you what happened after treatment interruption. On the left side is the vaccine group, on the right side is a placebo group, and each of the different lines represents the viral loads after treatment interruption. And this is a graph also showing the proportion or the percent of individuals who remained suppressed. So you start off at 100%, everyone was suppressed, and then over time, patients lost suppression, and so the line started going down. And what you can see is that the vaccine line is a red line, and the placebo line is a blue line. So you can see in this case, unfortunately, the vaccine, while in prior trials showed that it could boost the immune response, here, only with the treatment interruption trial could you see that it actually did not have a significant effect in terms of delaying viral rebound and leading to HIV remission, right? But I do want to point one thing out, is that you can see there are actually several individuals here showing here, both in the vaccine arm, but also in the placebo arm over here, who actually were able to remain suppressed, have the viral load remain suppressed without treatment. And these individuals are known as post-treatment controllers, people who can stop treatment, but yet keep their virus at a very low level. That is, it was a really interesting observation. Most of these individuals were treated during very early infection, so that may have something to do with it. But I think post-treatment controllers is actually a special interest of mine in my research. It is a kind of a different talk for a different day. I could talk a long time about it, but just to show that this trial actually found some really interesting results, even though the vaccine itself didn't have the effect that they were hoping for. So I do want to also at this point mention that these treatment interruption studies do carry some risk, right? And that we have to be cognizant of those risks as investigators and for our participants. And these risks include what's called retroviral rebound syndrome, which is similar to what patients might get during acute infection, some fevers, rash, having some swollen glands. Also drug resistance, as well as even there's been documented cases of HIV transmission during treatment interruption. Now, just to put these, give you a kind of a concrete example of these risks of treatment interruption trials, I'm showing you one particular study, and this is a study, again, that Raj has mentioned, of 2 patients seen at the Dana-Farber who had bone marrow transplants for cancer. Now, neither one of them received the donor cells that were resistant to HIV. They had regular kind of donor cells with regular CCR5. Both of these individuals ended up undergoing treatment interruption because, partly because, after the bone marrow transplant, couldn't detect any HIV. And both of these individuals ended up having treatment interruption. One ended up going had HIV rebound about 3 months after treatment interruption, the other one about 8 months, so for actually quite a long period of time. But both of them rebounded to very high levels. One individual, both of the individuals had some symptoms, including fevers and other symptoms, and one individual who was somewhat, non-adherent to the antiretroviral therapy ended up having a new drug resistance mutation, all right? So the risks are real. I will mention that these 2 participants represent 2 very special participants because they underwent bone marrow transplant, which means that their immune system had never seen HIV before. It was not their immune system, it was someone else's immune system, right? So this is what is actually seen during acute infection, when the immune system has never seen HIV before. And that is actually not what we see for most of the studies, kind of the studies on HIV remission, where we don't have such a dramatic effect. But I just wanted to demonstrate that risks can be present, although there's also ways that we can minimize risks. For example, minimizing risks of drug resistance. These individuals were on what's called an NNRTI regimen, and some of the newer drugs, some of the integrase inhibitors, have much fewer risks when it comes to developing new resistance mutations. And also in terms of not letting our patients be exposed to long-term high levels of virus. What we do is, at this point with the newer treatment interruption studies, once the virus is detectable, we immediately start them on antiretroviral therapy after they get to a certain point, so that we don't let them have long periods of viremia. So there are definitely ways to minimize the risks of treatment interruption trials. So, these trials, as I mentioned, do have risks, right? And because most of these interventions that we're testing probably are not, as of today, going to lead to an HIV cure, are these trials ethical? And when it comes to conducting ethical clinical research, we as investigators always try to abide by a certain set of principles. And in this case, one major principle is, does the research that we're doing have potential value to society and to helping future patients? Does the research that we're doing have a really strong and rigorous scientific validity, and that is built upon a foundation of good scientific research? And also, are we designing the trials appropriately? And we also have to really look at the risks and the benefits. Do the risks that we're asking our participants to take on, are they adequately weighed by the potential benefits, not just to the particular individual, but also to society and also to the future patients? And finally, I would say most importantly, informed consent. Do our participants really understand the research, the risks, and all of the potential kind of implications of the research? And especially since many of the research studies that are being done today may not have that immediate benefit to our participants. And I will say that these principles are the underlying foundation to all of even the early phase clinical trials that are done. Clinical trials that are performed in healthy patients, right? Not just in HIV, but in oncology and endocrinology, in any given field, and that there are I can give you a whole long list of medications that are being used today that were initially tested and studied in healthy individuals. And as long as I think that the research meets all of these ethical principles, it is thought that the research can be done and can be done ethically. And just to give you guys an example, this is a study that was published many years ago now on looking at drug-drug interactions between a hepatitis C drug called boceprevir, which is one of the first direct-acting hepatitis C drugs that came out, and raltegravir, which is an HIV medication, right? To see whether or not these two medicines have any unusual or severe drug-drug interactions. The researchers tested these two drugs on healthy individuals who had neither hep C nor HIV. All right, so they were not going to receive any benefit from this research. But what they showed was that when these individuals took both of these drugs together, the levels of both of these two drugs were excellent, were adequate, and that there really wasn't any drug-drug interactions. Because of this study, we had a blueprint in the clinic where we could now have a way to treat our HIV patients and to treat their hepatitis C in a safe manner. And none of this could have been possible without kind of a study in healthy individuals who were not going to benefit themselves, but there was going to be benefit to society and to other patients, okay? And so at this point, I'm just going to make a plug on one of my studies that we're working on through the AIDS Clinical Trials Group 5345. And the study is really focused on an observation that we've seen in prior studies. And those prior studies have shown that after treatment interruption, some patients will see their virus come back very quickly, within days. Whereas other patients, their virus won't come back for weeks, months, or even years, right? And why is it that that happens? And can we predict who might who might be what's called a long post-treatment control, or who might have more delayed viral rebound? And so this is as I said, can we find assays or biomarkers, what we call it, that can predict when HIV will rebound? And this is a study, ACTG 5345. Well, it has a long name, but really it's the ACTG kind of prospective biomarker study. And Just to give you an idea of why biomarkers are important in research, and not just in research, but in clinical care, I wanted to talk about HIV viral load. HIV viral load, we are all familiar with, is what we use to measure how effective our therapies are for our patients. But before 1997, HIV viral load wasn't available, really. And in order to determine whether or not a new HIV drug was active, we had to look at whether or not it affected the death rate of HIV patients, which made it really difficult to study. And then the FDA in 1997 looked at HIV viral load and said, this could actually be a better way of predicting whether or not a drug could work, because we could find that the viral load was correlated with clinical endpoints like death and AIDS-defining events. And also, if you change the viral load, if you change this biomarker, it could help improve the health and delay and decrease death. And so in 1997, the FDA introduced accelerated drug approval using viral load as the endpoint instead of some of these other clinical endpoints. And that really greatly expedited our ability to test new HIV medications and really ushered in a decade, I would say, of unprecedented expansion in the availability of HIV drugs. And now that we're turning our efforts to HIV cure, we need a similar biomarker, an HIV viral load for the HIV cure field. And so the study that we're doing is are enrolling individuals who between ages of 18 and 70 who have been on antiretroviral therapy for at least 24 months or 2 years, whose recent CD4 count is above 500, whose nadir CD4 count, or lowest CD4 count, was above 200, and who has no history of AIDS-defining illnesses. And individuals who are on an NNRTI must agree to switch to a integrase inhibitor-based regimen, that candidates must agree to use barrier protection for all sexual activity, and that a fully active alternative regimen must be available. We're enrolling 2 different cohorts, individuals who are treated during chronic infection, which is cohort A, as well as individuals who are treated during acute infection, or cohort B. And which patients might be a good fit to be a participant for 5345? Well, of course, individuals who are altruistic, want to contribute to a search for an HIV cure or remission, and are curious about how quickly HIV will return in the blood if they stop ART. Now, this trial itself does not have a vaccine or other intervention. We're really trying to see whether we can predict after someone stops therapy when the virus will come back. And also, there are some patients out there who I have spoken to who had already been contemplating pausing their antiretroviral therapy, and this gives them a structured way, a safe way as possible to do so. What are the potential risks, and how are we minimizing the risk? Well, in terms of minimizing the risks of CD4 loss, we restart antiretroviral therapy right when we see the viral loads reach 1,000 copies. So we do early ART reinitiation so that we don't allow our participants to have long periods of viremia. For the acute HIV rebound syndrome, our individuals undergo very close monitoring, twice weekly viral load monitoring, and prior studies have showed very low risk in this participant population. Drug resistance. So we ask our patients to switch off of NNRTI regimens to go on an integrase or a protease inhibitor-based regimen to decrease the risk of drug resistance. And then to prevent HIV transmission, we ask all of our participants to use barrier protection with all partners while they're on this study. So are there any benefits for the participants? There is not expected to be any health benefits for the participants, but the results, I think, could enhance our participants' knowledge about their own disease and satisfy at least some curiosity. And of course, it may help the HIV field in general in the search for an HIV remission and cure. And I think that through 5345, we hope to find assays that can predict when HIV will return after pausing antiretroviral therapy and can act as a signpost for researchers testing strategies for an HIV remission, potentially reduce the need for treatment interruption trials in the future, to assess any of the effects of these short-term antiretroviral therapy pauses, as well as to identify targets for HIV treatment, and overall really is to try to accelerate the development of strategies for HIV remission. So for anyone who is interested, the contact is Cheryl Keenan, and I have her contact phone number here, and then you can also email BWH Clinical Trials at partners.org, and I believe she is here and can talk to anyone who is interested. Again, BWH Clinical Trials at partners.org. So I'm just going to end it with a quote from Timothy Ray Brown, who says, "I know in my heart and soul that I will not be the only one cured of AIDS. Hope is alive in me," I think literally here. So thank you all very much. Happy to take questions. Yes. Question? Yes, go for it. Yes.
Okay. When you talk about HIV interruption, as I stated earlier, I'm somebody who's been over the age of 25 years.
Yep.
And I've actually had to stop interruption due to medical issues such as, some of the medicines I take, I cannot eat. So I can't take them unless I'm sorry. So they literally had to stop them for like 4 days because I could not take them. And then when I started them back up, I know there's in the past rather, there was a thing where if you stop your medicines like that, your body would get immune to those medicines. So this has happened to me multiple times. I can't take them. But still, when they do my viral load test, it still comes back undetected. And that's one of the things I think that you should talk about, the fact that what is the benefits and what is the risk when you have to medically stop taking this medicine. Can it make your body eventually become immune to it? And why is it for those persons who actually stop it, still their bodies actually go right back on it after 4 or 5 days? That's one question, and I want to do it real quick. And the other one, I think it's important that we talk about why is it so difficult for other people who are really interested in doing clinical trials. They will not qualify because of all of the guidelines that's set by, I believe, the FDA. Is that correct?
Yeah, so thank you for those questions. The first question had to do with stopping treatment, how risks and benefits in terms of will the body become immune to the to the drug in some ways. Our concern is less that the body will have some reaction or tolerance or immunity to the medication, but actually more has to do with the virus itself, especially with some of the older or some of the certain classes of antiretroviral drugs, like what I mentioned, NRTIs like efavirenz and rilpivirine, some of these drugs where when you pause the medications and give the virus a chance to replicate, you run a higher risk of developing drug resistance. Now, I think some of that risk can be minimized with some of the newer classes of medications like dolutegravir, bictegravir, some of these integrase inhibitors, where it's shown that even individuals who ended up having virologic failure, the risk of developing drug resistance is much lower. In terms of, you mentioned that you had stopped treatment for several days and the virus had not come back. And actually, I would say we expect that for most participants, the virus will come back, but not for several weeks. And as I mentioned, there are individuals who can be off therapy for months or even years, although those individuals are exceedingly rare. And they're a really kind of an important area of study. In terms of kind of benefits, as I tell all of my patients, don't stop your therapy, right? Unless you're part of a clinical trial, because unless you're doing it for a scientific reason, or unless you're having issues with your medications and there is some other clinical reason to stop your medication. If you're tolerating the medicine well and the medicine is working, don't stop your medicines right now, right? Don't do it on your own. And why is it that it is difficult sometimes for the patients and to enter into clinical trials, and why is it that the FDA has set such a high bar on some of these trials? I think sometimes appropriately so, right? Antiretroviral therapy at this point is relatively especially with the newer regimens, patients are doing well on it, right? They're living long, healthy lives, and we need to set a high bar to make sure that we're not, that the risks that we're asking our patients and our participants to undergo really is weighed by the potential benefits, and that we're not asking them to undergo too much risk. And that the reason why for our trial that we have certain criteria for CD4 counts, for example, is that we also want to make sure we're doing our clinical trial in the as safe a way as possible, that we have done studies in the past that show that if you had a really low CD4 nadir, which means that if your CD4, the lowest CD4 that you've ever had is particularly low, after you stop treatment, the decline in your CD4 count is much greater. So you run a much greater chance of having lower CD4 counts after you stop treatment. That's why we have a kind of a lower bar for what your CD4 nadir can be. So it was our way of trying to balance the risk and making sure that we select the participants who we feel are not at the highest risk in terms of entering these studies. So another question?
Yeah, so one of the things you mentioned in terms of the ethical issues and risks around conducting these studies are risk of HIV transmission. And so one of the things that I'm looking at with the testimony is bystander risk, and what responsibility do researchers have to partners and well, fetuses as well that have the risk that aren't aren't considered study participants, and you're not really responsible for doing informed consent and going through that process, but other than requiring clinic study participants to use barrier methods and inform them that yes, you will have higher viral loads. What is your responsibility, and what else can you do?
Great question. And this is an area of debate. Oh, yes. So the question is, what should be done about partners of participants who are undergoing treatment interruption and other people who might be affected by participants who are undergoing treatment interruption and potential transmission risk? And that is a great question. And that's actually a question that, an issue that we, within our study team, first of all, have really had a lot of discussions about, and had made it very clear in our in the study protocol and the consent forms that our participants really have to be using barrier protection while they're in the study to prevent transmission, because there have been reports of transmission to partners. And we have a pregnancy test during when the participants are enrolling. If there's any concern about pregnancy, they'd be taken off the study. And in this study, we believe that the duration that participants so actually taking a step back, the risk of HIV transmission is greatest in individuals with high viral load, right? And also individuals who have higher viral load for longer periods of time. In this study, once someone reaches 1,000 copies, so still a relatively low level, they are put back on antiretroviral therapy immediately. So the risk of this particular study, in terms of transmission, I would say we're trying to minimize that as much as possible. Having said that, there are other studies that are ongoing and that are being planned that might use a longer period of treatment interruption because they're looking for some specific immune response or some specific effect that requires a longer period of treatment interruption, right? And the risk in those situations of partner transmission is greater. And there is a lot of debate in the field about whether or not we need to provide what's called PrEP, for example, or pre-exposure prophylaxis to partners. What if someone has multiple partners? What if there's a new partner? Do they have to come in to be consented? It is a difficult question. People's lives are messy, right? They're not always with one partner, and they sometimes don't want that partner to know, or who knows, right? So and what is our responsibility? It is something that it is actively debated in the field, and we're seeking community input, and there's it's a thorny topic, I will say, right now. Yeah, question in the back.
I found it interesting, this first gentleman who asked the question why people who are HIV positive, who are willing to participate in options. And until you answered this question, I was wondering the same thing. I've never wanted to participate myself, but I do currently. But to your point is when I was diagnosed HIV positive, my CD4 count was 100.
Mm.
All of the other things. And for my CD4 count to go up to 300, past 300, it would be a little high. But when it came up just over 300, when I stopped being consistent with my medication for a period of 3 months, and within that period of months, it went down to 148. And the challenge I found after getting back consistently adhering to my medication was that my immune system was so spent that I would get sick for all the regular things that people who have healthy immune systems don't have to worry about. And so having to relive that over again was exhausting.
Yeah.
I thought one of the challenges everyday living was. So I understand that, if it's working, you tolerate the side effects, that don't stop. And it's so true in my case.
Mm.
It's still, I haven't been able to reach a really high level of CD4, and I wonder if it's just that my reservoir I'm at a point where my reservoir is just, that's it. And so I often wonder, why can't I hear people, who've been on antibiotics for years and years, and CD4 count went to the single digits, and then they're over like 800. I'm like, what's wrong with me? And it's just hearing it medically in this format. I feel like a load of wondering, like, what's wrong with me? Why can't I understand or get it? And this research makes it so much clearer in the way you articulate it. I feel like a burden has been lifted in terms of, I feel now that I'm part of the conversation.
Thank
Part of
you.
the conversation. I get it. I'm not the smartest guy, but I'm not dummy. I feel so healed. I'm so great. I feel like I'm so glad I got my
Well, thank you for sharing that story, and I really applaud you and everyone here who are interested in participating in research. And there are lots of different ways, different studies that you can participate in, especially ones that don't involve a treatment interruption. But there's a lot of ways to help out with this search for a cure. And I think, first of all, your story is not uncommon in terms of the immune system. Once it gets down to a low level, has a hard time bouncing back to the absolute highest level again. Even though it has recovered, and even though CD4 count may not be in the 800s or 1000s, your immune system still is enough to protect you, against almost all the diseases out there. But it may not reach the highest level, and that is absolutely a common thing, that a scenario that happens. So you are far from alone out there with that. But thank you so much for sharing that story with us. So, yes.
My question goes back to the early patient, and in your comments about the reservoir that's below whatever you measure the rise in the blood at. So my question actually is a second part of not really clear, but first I want to know what happened to his reservoir? Did he have a reservoir? And was the new cells in his bone marrow enough to overcome the reservoir? And then for me, I remember when that story came out, thinking like, yeah, like I want a bone marrow transplant.
Yeah, thank you. So the question was about the Berlin patient, what happened to him, what happened to his reservoir. I mean, the Berlin patient really, he was a very special case, one that has not yet been replicated, right? But his case has really supercharged that area of research and that field as a whole, and really inspired, I would say, all of us. His reservoir, most of the reservoir is within CD4 cells, right? In order to clear his cancer, they had to try to kill as many of his bone marrow and the CD4 cells as possible. So he underwent pretty toxic chemotherapy and whole body radiation therapy to kill all of his immune cells. Even then, not all of his immune cells died because his cancer actually came back once, even after the bone marrow transplant, right? He had needed 2 transplants in order to finally get rid of the cancer, right? And I think it was probably a combination of the chemotherapy, the radiation, and getting cells that were just resistant to HIV. Now, one thing I didn't mention is that the donor cells doesn't just come in and just come in and take over the body, but it also recognizes some of the host's immune cells as foreign. And so there is something called a graft versus host effect, where the immune cells that he's getting from the donor is all of a sudden starts to kill his own immune cells. But that's a good thing, because his own immune cells are cancerous. So that was an added way that I think he was able to kill the remaining few cells that were left over after the chemotherapy and the radiation therapy. But so again, it is a highly toxic regimen, but I think all of those things combined did it.
Check on them. Why were the donor cells in any way valuable, like a heavy lax? I mean, does that donor, does that have something unique in their blood?
Well, yeah, the donor cell was, it was lacking. Well, it was lacking in CCR5, right? So that donor cell had a deletion in the CCR5 gene, right? And so so even if there were some host or some cells of his own left over that were producing virus, it couldn't infect any of the new cells that came along. Now, remember how I mentioned the 2 Boston patients who also received bone marrow transplants? Their cells were not resistant. The donor cells were not resistant to HIV. And both of those individuals, their virus came back.
That wasn't the donor cells that worked like that, used like they did Henrietta Lacks.
When you say use like the cells from Henrietta Lacks, as in why couldn't they be cultured and given to other people and things like that? Because if we receive cells from someone else, our body with our intact immune cells would instantly reject those cells and kill all the cells that were just coming in. His body didn't do that because his immune cells were gone, were already removed by the chemotherapy and the radiation. So, and if he didn't have chemotherapy and radiation, his body would have also rejected those cells. So to have a successful bone marrow transplant, you have to have the really toxic treatments first before you can successfully engraft with the donor cells. That's why just injecting cells from Timothy Ray Brown to our patients now would not work without the other part of it. And the other thing too is that the CCR5 deleted cells, individuals out there with this particular CCR5 deletion are actually pretty rare. It's found in very few individuals. So he was very lucky in many ways to have found a donor who had enough what's called HLA matching. So the donor was somewhat, had a similar kind of markers to his own body, so that he could be a transplant candidate, a donor candidate, yet also have a CCR5 deletion. So he was very lucky to found that particular donor that matched all of the criteria that is needed to receive those cells and to have the deletion as well. Yes, question?
You mentioned one of the benefits being for the participants to learn about their HIV
Mm-hmm.
disease. I'm wondering if there's been any research or results about the timing of viral rebound for later interruptions in their retroviral therapy. If there's some kind of correlation that says if you were
Yeah, we don't know. So as in, if you rebounded once, what happens the next time you rebounded? There's not enough data. There's very few. There is not enough research done on that I can even hazard a guess at this point. Yeah. So.
So there's no way to predict if what do you call them, the PICs?
The PTCs, post-treatment controllers, right?
Would they always be?
Will they always be post-treatment controllers? We don't know. We don't know. These individuals are so rare. I'll just give you an example of how rare post-treatment controllers are. We have we've looked through all of the prior AIDS clinical trials group studies. We've looked through cohorts from the Reagan Institute, from University of Washington, from UCSF, from University of Montreal, from University of California, San Diego. All of them have various cohorts of patients. We found maybe less than 100 post-treatment controllers out of thousands upon thousands of individuals who've undergone treatment. These are very rare individuals, I would say, overall. So, yeah. Oh, I have another question. Actually, one more. Sorry, one more question.
How much time, first of all, why didn't it work the first time? How much time did lapse between the first procedure to the second procedure? And why did it work the second time and not the first time?
Yeah, so what do you mean, why did the cancer come back?
The German guy.
Right, the Berlin patient, right?
He had it twice.
He had it twice.
The first time it didn't work. How come it didn't work?
Yep.
And the second time, how long was it between the first and the second?
Yep.
And why did it work the second time?
Yeah. So why didn't it work the first time? I don't know. What I'll tell you, when I say it didn't work, what I meant was that he did the treatments, got the infusion, and then cancer came back, right? So it meant that there were some cancer cells that were not fully killed by the regimen that he was on, and the donor cells, maybe not enough of them engrafted for some reason, such that there was room for the cancer cells to come back. And then when they did it again, the whole thing again,
How long?
I don't recall off the top of my head how long that was. It wasn't long. It was weeks or months. It wasn't a long years and years. It was within weeks or months. So all right. Anyway, thank you all.